Um, so I could like to give a talk about antioxidant and cytotoxicity activities of intracellular polysaccharides extracted from Physarym polycephalum microplasmodia. Sorry, I left that out. Uh, this is the outline of the talk. So I will start with the introduction on microbial polysaccharides. Um, Polysaccharides are polymers which consist of monosaccharides or sugars. And there are three main types of polysaccharides found in microorganisms, depending on their position and also their structure, which include polysaccharide in the cell wall or cell membrane, or structural polysaccharides. And also microorganisms can produce exopolysaccharide, meaning the polysaccharides which are excreted outside of the microbial cells. And the third type is intracellular polysaccharides, which are produced inside or within the cell of microorganisms. And polysaccharides from microorganisms, especially from fungi, have numerous biological activities, including antioxidant, antimicrobial, anti-tumor activities. The mechanisms of anti-tumor activities could be quite complex. Sometimes the polysaccharides can boost the immune response of the bodies instead of killing the cancer cells directly. So I am also would like to introduce a little bit or some feature of Physarym polycephalum before I go to previous studies on myxomyces polysaccharides. So you are familiar with the fruiting bodies of Slamos, of course, and plus their plasmodia. But the mycoplasmodia are a bit different. So plasmodia, when being cultured in liquid medium, can be fragmented into mycoplasmodia. And so far we have been able to culture only mycoplasmodia of Physarym polycephalum under laboratory condition, even at a fermenter scale. Next is I would like to introduce some of our research group on polysaccharide from plasmodia. So here is a plasmodium culture. And one of the reasons why Slamos got the name Slamos, in my opinion, because they normally leave a slam check behind when they migrate from one area to another. So we investigated the chemical composition of the slam checks, and also the bioactivity of the slam checks. Besides that, we also extracted exopolysaccharide of the plasmodium of two species, Physarym polycephalum and Physarym oblonga. These two can be cultured quite easily under laboratory condition, as you already knew. So for the exopolysaccharide of the plasmodium, you get to carefully collect the plasmodium and put it in saline water or sterile water and gently stir it, and then remove the plasmodium by a gentle centrifugation. The point is that we don't want to break the plasmodium because the intracellular polysaccharide could be released out. And then we collect the exopolysaccharide, and I'll also do the same with the SLAM check. And now the data show that the SLAM check actually don't have both species don't have significant biological activities. Probably because the function of the SLAM checks would be just simply to mark the area that has been already exploited by the Plasmodium. However, the EPS or the exopolysaccharides have a significant one species of Oblonga have the significant antifungal activities against this Candida albicans. And also we tested their abilities to kill some cancer cell lines, including breast cancer and liver cancer cell lines. And this data is quite interesting. They could inhibit 50% of the cell populations at the concentration around 1 milligram per mil. Taken in consideration that these samples are crude, so they are very low compared to positive control, but they are crude samples, so they are somewhat promising. So this is the very first research in exopolysaccharide or polysaccharides in general from SLAMOS. And then we did another one with the intracellular polysaccharide of two species. So for this one, we collected the Plasmodium. These are just for illustration, right? We collected the Plasmodium and extracted out the polysaccharides, and we also removed the unbound proteins and other impurities from, we call the partial purification of the sample. And because the monomers and also the size of the polysaccharides are important to their biological activities, they actually are thought to be linked with the biological activities of the polysaccharide. So we analyze the monomer and also the molecular weight. And obviously the intracellular polysaccharide of two different species have a different monomer composition and also molecular weight. And also we found that intracellular polysaccharides of Oblonga had stronger antioxidant activity. But that of polycephalum had higher anti or cytotoxicity activities against HeLa cell or cervical cancer cell lines. Also, the IC50 is around. No, this one can kill like more than about 70% or 67% of the HeLa cells at this concentration. It should be noted that there is no significant difference between the crude and the partially purified samples in this case. So the next research dealt with the microplasmodial culture of Physarym polycephalum alone. So for this one, we centrifuged to collect the mycoplasmodia, and then we extracted the intracellular polysaccharide from the mycoplasmodia. And also for the supernatant, we collected exopolysaccharide from the supernatant. And also we try to remove proteins, unbound proteins from both the EPS and the IPS of the sample. And we found that for the removal of proteins could influence the cytotoxicities of the samples, but not in the same manner. For example, for the HeLa cells, the cervical cancer cell line, the IPS, the partially purified IPS, had a better activity compared to the crude one, but it's not the case with the breast cancer cell line. So, and also we went a little bit deeper to initially analyze the mechanisms of how these samples inhibited the cancer cells, and we found that it's most likely linked to apoptosis. You can find more information of this in this publication. So going back to this current research, for this this research, we purify the mycoplasmodia crude IPS, intracellular polysaccharide, and then we screened for the fractions with high carbohydrate contents, meaning like they are most likely to be so because of the this is not really necessary if you have a lot of time and funding. But if you go with the high carbohydrate contents, it means you can obtain sufficient amounts of samples for the biological evaluation. But it's not a fixed criteria. And then we evaluated antioxidant and antiproliferative or cytotoxicity of the fractions, the IPF fraction against cancer cells. And this is the procedures, research procedures. And now, so first of all, the U of the IPS form of the plasmodium first to give you some idea of how much of the sample we can obtain. We count 1 liter of the sample of the culture medium can give around this amount of mycoplasmodia. And from this, from 1 gram of the mycoplasmodia, we can get about 0.2 gram of the crude IPS. And then we use iron chromatogram to purify the sample. And there are 3 potential fractions based on their carbohydrate contents and the amount. We named F03 and 4 and 6 based on the concentration of sodium chloride used. And these are the obtained fractions was concentrated dialyzed to remove the salt and then freeze dried. And then we weigh the weight of the fractions, and then this just shows the rough estimated percentage of the fraction in the crude IPS. And then we evaluated the antioxidant activities, and they definitely have different activities, antioxidant activities, and the F03 had the highest activities among the 3. And then we calculated the IC50. So even though the F03 had the higher antioxidant activity compared to the other 2, however, the activity is still much lower compared to the positive control, which is ascorbic acid. And then we evaluated the cytotoxicity of the samples against the breast cancer and cervical cancer cell. And this of different concentrations of 3 fractions, and the control has just a sterile buffer, and CPT is a positive control. The concentration is super low. It's 1 microgram per ml. So you can see. And and for this one is the data of the fraction against breast cancer And for the one on B, the figure B is against hair cancer cell. And in both cases, you can see the fraction F06 have had a better activities. And we also tested the toxicity of the fractions against the normal cell lines to see if they have they kill the normal cell lines or not. This is the skin cell line. And now, we tested with only the fraction number 06. And at different concentration, you also the toxicity of the F06 to the normal cells are much lower. Even especially at the concentration. This is a bit weird, but at higher concentration, the toxicity is lower. I think they are not significantly different. So through these two data sets, we putatively conclude that the F06 fraction is toxic to cancer cells, but quite safe with the skin cell, and we are working further with this fraction. Yes, that's all of my talk. And so I would like to thank some organizations for funding the research, and I also would like to thank my former supervisor, Professor Stevenson. He introduced me to the world of slummers, and he has supported our research group immensely. And I'm also lucky because I have not many, but several students who are very devoted to their research. And this the research I just presented was actually done or carried out by by my undergraduate student, and some other aspects of the research are being carried out by my graduate students. And thank you for listening.
That's very interesting talk. Do we have any questions?
Thank you for the interesting talk. I have two questions. First, I was surprised to hear this hypothesis that slime tracks, slime remnants, can be used by Plasmodium to mark the exhausted territory where it already have eaten everything. It sounds very smart from the side of Plasmodium, and sounds very reasonable from the evolutionary perspective. But do you know any proofs, any experiments that prove this supposition?
I should have cited some reference for that. Actually, it's just one of the possibilities, and I don't think I am because I had to think a little bit whether in that paper is not of our group, of other research group, and let me try to recall if they actually did some experiment or it's just like a proposed explanation. I can't think of, but if I have to to check that hypothesis, I could just culture I could just cut a piece of actively growing Plasmodium and put it maybe at the end of one slime check to see if it could be interested to travel again through that check.
Yeah.
I'm
Exactly.
sorry, if I find that paper, I will send it to you, or I will.
So this hypothesis seems to be rather easily checked. Okay, and my second question, is it possible to use your methodology to identify polysaccharides inside mature fruiting bodies in collections? And how much material do you need for this? So can you identify polysaccharides not only in Microplasmodium, but in mature fruiting body, in stalks and peridia? Is it possible with your methodology?
For fruiting bodies,
Yeah.
Fruiting bodies that first of all, fruiting bodies would contain polysaccharides too, but we need normally we to carry out the 2-hour research, including the chemical characterization and also biological evaluations. You need quite a lot of samples. So, but I can't tell
So
how
amount must be pretty big, right?
Yes. So for the fruiting bodies, first of all, they are too small, and we never tried to work with them before.
So if I give you 5 fruiting bodies of Physarym,
Oh, no.
you cannot do it.
No.
Okay,
Maybe
it's a pity.
But actually, we are looking for some other compounds from fruiting bodies of Stamos, including stamonitis. So if you kindly offer me stamonitis and furigo, I would be very happy to accept. We got another project which is very different from polysaccharides.
Thank you for your presentation, but I have one question. What can explain the greater viability of HeLa cells at a higher concentration of IPS?
Oh, that's a good question. This one.
No, or previous. Yes.
Yeah. So for the
1,000 nanograms per milliliter.
yes, this is when it's actually they are not significantly different, huh? I think according to the statistical levels here, it is more. So here, what with the activity is not dose dependent. Maybe just because I mean maybe this would be the highest activity the sample could get, or another possibility is that this sample is not really completely pure. So what we are doing is that we try to determine if there is something else in the sample or just only one kind of polysaccharide, and then we will re-evaluate the bioactivities again, and maybe then after that the activities could be different. Yeah.
Okay, and one more question. Did you analyze possible practical application of this IPC, or just study their properties?
Oh, it depends on the outcome of the data. So if they are really significant, the activities are significant, we would go further.
Do you have any more questions? No? If not, then thank you so much.
Thank you.